Those are very nice presentations. I don't really have data to show you, so consider this more of like a work in progress. So yes, I titled the talk Innovating the Use of Repellents Beyond the Siegel Archetype, and sort of just want to discuss like how we get into this line of thought. So I originally started in Steve's lab. I worked with Pat doing just basic mouse semantics that were effective. And then I was looking for a reason to stay in the lab, and I was hired to continue a DoD project, which looked to see if we can apply the use of spatial repellents to ticks. And this is sort of a new idea for ticks, because the main reason that we use spatial repellents is for insects, and that induces either an excital repellency response or some kind of vaporized response that either deters mosquitoes from landing. But the main thing is they're not actually touching the insect itself. They're sort of perceiving the vapor that's. And so one of the challenges to actually applying spatial repellency in ticks was developing an assay to determine whether it's effective. Ticks do not behave like mosquitoes. They do not behave as you predict. That's been sort of one of the main challenges. How do we measure this repellency, and how can we apply it? And sort of taking this step back, thinking about a bigger picture of how to use repellents. If we look at the process by which a a tick, as it emerges from leaf litter, it goes through the host seeking process from the beginning to when it gets unfolded. It comes out of the leaf litter, and there's a series of interactions, whether it's as it's questing, the interaction between when it comes to your host, and then once it goes onto the host, and then a series of smaller behaviors before it gets being molten. One of our ideas is to actually define these behaviors, define these checkpoints that a tick has to cross as it goes through this life cycle, and see if there's a way that we can manipulate these behaviors with repellents. Sort of as almost a substitute for the synergism effect with the parasites, and see if there's a way that we can use some of the molecules that we've been developing that either show effects based on a tick won't cross from point A to point B. Can we apply that to sort of non-infections? And so this sort of line of thought is taking us from, think of these ideas of how we can use repellents in other than just spraying either skin or treating clothing, find these behaviors that a tick needs to go through, translate those behaviors into quantitative measures, standardize the assays that are used to assess these measures, and then we can sort of piece together this algorithm of we test a molecule with assays A and B, and based on the results of those assays, those could translate into different specific use cases. One of the molecules that we've worked with is NEWVEC, which Thomas worked with as well. And in the lab, what we see is it was originally thought of as a lethal molecule. So it kills ticks at very low concentrations. And at concentrations, I've only gone as low as 0.0029% in the lab. And this is with adults. The ticks actually won't stay on a filter paper that's treated long enough to induce a lethal effect. So the ticks are being repelled at an even lower concentration. And one of the reasons that this is meaningful is there's a study where comparing emulsified UP2 formulations with encapsulated UP2 formulations. And if you treat an environment with encapsulated UP2, the molecule actually persists in the environment for several months at roughly the same concentrations that it was treated at. And the phenomenon that you see is when you spray the environment and you flag the environment, you get almost no ticks, if any. And the question is the effect that we're seeing, the ticks that are there are being killed, the ticks that are coming onto this sprayed environment are also being killed, or are the new ticks that would be colonizing this environment being repelled before they can get there? And so the importance of understanding that is if that is the case, then that's justification for the use of environmental measures and wondering whether you can either use these dual action molecules for that role or use these molecules in combination with lethal effects. And sort of this extends beyond that too. So that could affect the questing stage. But what about like on-host measures? So in for tsetse flies, there's a very effective collar that was developed that emits spatial repellents that actually prevents tsetse flies from causing disease in cattle. And so that works with the phenomenon of using non-host semiochemicals. So pretty much masking the scent of a good host for a tick with the sense of a bad host for a tick that the tick perceives and then that host. And so this has actually been applied very briefly in dogs using some vertebrate-limited alamos against brown dog ticks. So another question is, can we take that phenomenon, use these non-host semi-chemicals in either as spatial repellents or contact repellents, either treat reproductive hosts or treat environments with it? Can they be applied to fabrics, either treated fabrics or manipulate fabrics that are impregnated with them? And so different use cases, the idea is take a molecule, see where it fits into affecting the tick's host life cycle, and then find use cases for those molecules. And so there's a number of other questions that we can ask kind of like based off of this. So in some of the blood meal analyses, what we see is that ticks are feeding and that they're dropping off and finding new hosts if you're being groomed off, or they're falling off. And so some of the papers we're seeing are 5 to 10% of ticks are having these incomplete blood meals that have DNA from 2 different hosts. And one of the reasons that this is important is we actually need to figure out if we were to use repellents, we're not killing the tick, so it could fall off whatever host or whatever surface. How likely is it that that tick can actually find a second blood meal? How efficient are ticks at actually doing that? Because if a tick that is repelled 5 to 10% of the time can actually find another untreated host, say it's like a reproductive host, the maximum efficacy that we would be able to get from sort of trying to fill in the role of the parasite would be 5 to 10% lower than the maximum efficacy of the comparable molecule. And so that's an important question to answer. Are we actually seeing 5 to 10%, or is that a little bit inflated either based on some of the DNA analyses that are not efficient? And another thing to look at is one of the, I guess, qualities that you would look for in a repellent that might be better than the parasite is that you lose the selection for resistance that you might see with parasites. So if we can substitute, especially with like cow ticks in certain areas, if we can find these molecules that work for either treating environments or treating some of these animals with these repellents, what do the effects look like in terms of sustainability? So that's what I want to discuss. So we're kind of piecing together this pipeline of defining the behaviors that a tick has to go through from emerging out of leaf litter to finding a host. What behaviors can we manipulate? How effective are these molecules at actually manipulating them? And then kind of set the stage for like kind of poking the system with different environmental conditions. So can we actually get repellents to work and apply them? But what happens in the sense of like heat waves? Are the repellents less effective because ticks may be more desperate in heat waves, or like climate predictions? And so trying to figure out system. That's all I wanted to discuss today, but hopefully in the next couple of months we'll have some actual data.